I'm giving an overview of brain tumor diagnostics and classification, looking back at how the World Health Organization has done this over the past approximately 50 years, talking about the recent 2016 WHO classification of brain tumors, then talking about the response that the community has had to the 2016 classification. And finally, for most of the talk, I'll be discussing the future of diagnostics as an integrated specialty. Until recently, until the 2016 WHO classification, they were classified nearly exclusively on the basis of light microscopic techniques, such as standard light microscopy as well as immunohistochemistry. It became clear over the 1990s and first decade of this century that molecular genetics could play a role in the classification of these tumors in addition to light microscopy. And the 2016 classification introduced molecular parameters in addition to microscopic parameters as the bases for brain tumor classification. For many of the major tumor types, and by major tumor types I mean the most common ones such as the diffuse gliomas, which are the most common brain tumor types of adults, as well as the most common malignant tumors of childhood, the Medulloblastomas, we introduced genetic parameters as a way of classifying the tumors over and above the histological or microscopic parameters for classification. So it affected a good number of the brain tumors, whether you had to include genetic analyses in their classification. For the diffuse gliomas, which are the most common types in adults, the most important genetic parameters are mutations in the IDH or isocitrate dehydrogenase genes. Those are IDH1 and IDH2, and those are used to classify all of the types of diffuse glioma, such as glioblastomas, astrocytomas, and oligodendrogliomas. The other key parameter is the copy number status of chromosomal arms 1p and 19q, and those changes are essential in the classification of oligodendroglial tumors. For medulloblastomas, there's a broader array of alterations that are used in their classification. And how one does it in medulloblastomas, whether one uses RNA-based approaches or DNA-based approaches or even protein-based approaches, is still being debated by the field. Nonetheless, by identifying specific pathways, such as the Wnt pathway and the sonic hedgehog pathway, you can divide medulloblastomas as well. It's, at the present time, it's mostly helped assign tumors into much more specific categories that then can be treated in a much more uniform manner. It has also helped assign tumors into categories for clinical trials in which one can use molecularly targeted therapies against the aberrant genetic abnormalities. These, however, have mostly been in the setting of clinical trials. There is also the practical step in the tumor known as glioblastoma, which is the most common brain tumor, of testing for MGMT promoter methylation. That's methylguanine methyltransferase. This is a gene, when its promoter is methylated, it has prognostic and what we call predictive importance. Predictive meaning that it predicts the response to therapies. And it's clear that if a glioblastoma has its MGMT promoter methylated, it will show better responses to the standard therapeutic approaches at the present time. When you look over how technology has impacted diagnostics in the last 100 years, you note that each time a newfangled idea comes in, 100 years ago it was light microscopy. In the 1960s it was electron microscopy. In the 1980s and 90s it was immunohistochemistry. In the 1990s and then 2000s, it was PCR, and then DNA sequencing, and then RNA expression profiling. Each time one of these comes in, it's a big impact on the field. People get very excited about it, and you spend a while figuring out what is the exact situation that new technology will be used in a practical manner. The most recent technology to make a big splash has been methylome profiling, a very important technology, and again, that will make an impact in specific situations. The question for the future, however, is not necessarily will there be new technologies that make an impact. The answer to that is yes. But how does one take multiple different methodologies and integrate them together to produce a diagnostic estimate that's more accurate, more predictive, more prognostically accurate than any individual one? And so what we're looking at now is the use of larger data sets, using disparate types of data input, not only the type of data that one can find from looking through a microscope or doing a methylome profile, but also the kinds of information that can come from a patient's blood samples or a patient's background DNA from their genetics, potentially in the future even their microbiome. But there are multiple different data sources, and so what we're exploring at the present time is the use of computational methods to integrate those many different types of data sources to produce a more accurate diagnosis. Sure, in brain tumor research there is greater international collaboration. This has been the case for a couple of decades in the pediatric realm, in which pediatric brain tumors are relatively uncommon, and so in order to make progress, the pediatric brain tumor community has done a very good job of collaborating internationally to move the field forward. It's less common in the large scale, the more common brain tumors, such as the adult brain tumors. But what we've seen over the years, most recent years, is a move towards greater collaboration, greater national and international efforts. One example being the Cancer Genome Atlas, pushed forward by the National Cancer Institute in the USA. The diagnosis of brain tumors has gotten much better over the last couple of decades. Gotten more accurate in terms of prognosis and prediction. It has a long way to go, and probably through integrating larger data sets, we'll make further progress.